We're happy to have here a holiday edition of the Stanford Wearable Electronics Initiative seminar. And our topic today is around flexible electronics for stem cell research, as well as brain-machine interfaces. We're very happy to have a special guest lecturer today, Dr. Jia Liu. And eWare was founded by Professor Zhenan Bao to foster multidisciplinary collaboration between Stanford University and companies. And we're very happy to have participation from more than 80 research groups across the School of Medicine and the School of Engineering, as well as grateful to our member companies for their support, which makes seminars like this possible. So for folks online, our program manager, Katrina Dillard, is hosting this seminar on Zoom, and you'll have an opportunity to ask questions of our speaker after the talk. So please type in your questions on Zoom into the Q&A. So the title of our lecture today is Soft and Flexible Electronics for Stem Cell Engineering and Brain-Machine Interfaces. It is my pleasure to introduce Dr. Jia Liu. Dr. Liu received his PhD in chemistry from Harvard in 2014, after which he completed a postdoctoral research position here in Stanford with the Bao Group. He joined the faculty at the Harvard School of Engineering and Applied Sciences as an assistant professor in 2019. And at Harvard, Dr. Liu's lab focuses on the development of soft bioelectronics, cyborg engineering, and computational tools. Dr. Liu is recognized for his innovations in soft electronic materials and nanoelectronic architectures for tissue-like electronics and their applications in brain-machine interfaces, high-density cardiac mapping, and stem cell maturation. He has received awards for his pioneering research, and a few of them are: he received the Inventors Under 35 by MIT Technology Review, the Young Investigator Program Award from the Air Force Office of Scientific Research, and the Catalyst Award from the National Institutes of Health Directors Pioneer Award Program. Dr. Liu is also an entrepreneur and is co-founder and scientific advisor for the brain-machine interface company, Axoft Inc. So welcome, Dr. Jia Liu.
Thank you very much, Angela, for your introduction. And thank Zhenan for inviting me to give a talk here. And it has been like 5 years since I left Stanford, and it's my first time come back to give a talk and kind of like a report to Zhenan and the Bao Group about what I have done in the past 5 years. And please feel free to ask any questions, like if you have, like during my talk. Very happy to address them. So my research topic today is soft and flexible bioelectronics for the stem cell engineering and the brain-machine interface. So my lab at Harvard, we are very interested in understand how the neurons in the brain function to give rise to certain behavior, how the malfunction of the cell give rise to the disease, or how the different type of cell assemble together into a piece of a functional tissue. And be more specific, like we are very interested to develop the platform technology that convert the high performance nanoelectronics into the flexible and the soft bioelectronics for the tissue interface. Integrated that with the technology, such as spatial transcriptomics, to look at the cell property from the to address those questions. Before I discuss before I discuss the technology platform that we are building, I want to first talk about why we want to build the technology in this way. Use the brain-computer interface as an example. So if we want to build an effective brain-computer interface to comprehensively understand the function of the brain, there are certain challenges that we need to address. The first challenging that is really the single cell activity across the entire 3D brain really matters. So unlike the computer chips, like a brain function in a way that it really recruit the cell activity at a single cell level across the entire 3D volume of the brain. So on the left, that is a functional MRI imaging of a human patient. And those red color label the region that where the neuron is under firing. You can see this is a patient that really need to recruit its cell activity across the 3D brain tissue to define its patient's brain internal state. And on the right, that will be the calcium imaging of a zebrafish larva. That is a transparent species. And each dot basically label one neuron that is under firing. You can also say that this fish need to recruit the neurons that are distributed across a 3D brain to define its like a behavior state. But if we want to capture those cellular activity at a single cell resolution across a 3D tissue, then we will face the second challenge in the neuroengineering that calls the spatial temporal dilemma. So those like each individual neural activity, this action potential, we call it one spike that only lasts for 2 to 3 milliseconds, confined into the individual neurons at a spatial scale from 10 to 100 micrometer. But we all know that the brain does not operate at a millisecond time scale, right? It takes days, months, or even years to build up the cognition, memory, emotions, and recruit like millions to billions of the cell at the spatial scale from centimeter to even meters. And for the different like species, like from frog to human, you have like millions of the neurons to like billions of the cells. That ultimately the bandwidth of the technology that we need to build requires the bandwidth around the hundreds of gigabytes per second to even thousands of terabytes per second. Then as a direct comparison, that a 720p movie that you watch on the YouTube, that is only 2.5 megabytes per second. So there's a huge gap between the technology that we have right now to the technology actually we want to build in the future. And for those like biological system, like a brain, the different cell will have the different gene expression, they have the different morphology and connectivity. And those cell type specific information will give rise to the different electrical behavior. So ultimately, we also need to integrate the cell type specific information with our like a technological platform. So, but why we think right now is really the golden era for us to address those questions, because driven by the Moore's law, on one hand, the number of the transistor on our each individual chip already goes to the number that is equally to or even higher than the number of neurons in the human brain. And on the other side, those like nanotechnology already miniaturize the sensors and the stimulators down to the subcellular size that in the in vitro settings can long-term stably probe those each individual cell activity. So on the one side, that is our like human brain, billions of neurons. On the other side is like a powerful computer chip, like billions of the transistors. What we are missing right now is really a cable, a scalable interfacing technology that can bridge those electrochemical impulses in our human brain to the powerful hardware to decode the activity. So what is required for this kind of a scalable interfacing technology? That will be summarized here. The X and the Y, that is the time and the spatial scale. And these two dashed lines label the resolution, the single cell, single spike spatial temporal resolution. And there's a third dimension that is a cell type specific information need to be incorporated into this platform technology. And what is like a current conventional technology that can do? Those bioelectronics can easily achieve the single cell, single spike spatial temporal resolution. But their performance in the biological system cannot last for a long time. They cannot be easily distributed across a 3D volume of the tissue. And certainly there's no method that can incorporate the cell type specific information into the platform. So since we already have such like a powerful semiconductor manufacturing technology that is enabled by Moore's law, why not we just use that technology to build those high density electronics and probe with the brain? So this is a very famous technology that already pursue in this direction that called the neuropixel. I'm sure like most of people know that, right? It's just using the CMOS fabrication technology, integrate thousands to like 10,000 of the sensor together, make those like miniaturized silicon probe and get them into the brain. Well, those technology allow this like thousands of the neurons recording at the same time, but their stability is very limited. Like in the very beginning, you can look at those like, you can capture those thousands of neural activity, but those signals are gonna quickly decay. That over the time, you will lose the recording capability from those neurons in the brain. So in the first part of my talk is really want to develop this tissue-like nanoelectronics that enable the long-term stable electrophysiology at the single cell resolution. But then we want to understand what is really prevent us from getting this kind of long-term stable interface with each individual neurons in the brain. Turns out it's really coming from this size and mechanical mismatch between the brain and the electronics. So brain is very soft like a tofu, and the electronics on the other hand is very rigid like a blade. So if you put the electronics into the brain and the brain is constantly moving, it will cause, first is the constant drift of the probe inside the brain. The probe will cut the brain at the micrometer scale. That even you can record the neural activity, every day you record it from like a completely different neurons. And then it will trigger this huge immune response from the brain tissue to the implantable probes that over the time at the implantation side, you will have a degradation of the neuron, you have a proliferation of the immune cells, so that you will lose the single unit action potential that you can record in the very beginning. And for the patients with the deep brain stimulators implanted, every couple of months of a year, you need to relocate the stimulation location to find a healthy neuron to do the stimulation. So when we look at these questions like a long time ago, we are thinking like if you look at the history of the mankind, have we developed any kind of material system that can bypass this size and mechanical mismatch with the biological system, provide this long-term stable interface with the biological system? Turns out we actually already have that. So in the tissue engineering or regenerative medicine, people have developed this so-called synthetic tissue scaffold, which is a three-dimensional microporous material that can support the cell growth and then transplant back into the human body without trigger this severe immune response. So we are thinking, why not we build the electronics to mimic their structure? They also have this 3D microporous structure, subcellular feature size, and tissue level mechanical property. But most importantly, is that those like material, those structure need to be made by the state-of-the-art lithographic technology so that it can be scalable. That means like hundreds of the electrodes that we made in the lab can directly translate into the semiconductor industry to build like millions of them. But in the very beginning, actually, we cannot even make the tissue level soft electronics at all, because for all the material that is used for the nanofabrication, like SU8, polyimide, gold, silicon, they have the Young's modulus at 6 order magnitude larger than that of biological tissues. Then while we have some like a soft material at that time, they are not compatible with the lithographic fabrication or the bioelectronics at all. So in the very beginning, we can only make the tissue level flexible electronics because the flexibility is not only determined by the Young's modulus, but also determined by the thickness of the device. We can use those rigid material, make a very thin electronics so that they can have the tissue level flexibility. So this is almost like 10 years ago that we first published this like a flexible mesh electronics that is only 800 SU8 sandwich like a 20 nanometer gold and in the middle. And they made it into this like mesh-like structure so you can see how flexible they are. They can even like inject into the brain through the syringe injection. And over the year, we can further improve this like implantation side, incorporate those like micro needle user for those kind of mesh electronics implantation. And across like implant them across multiple brain regions. The green color that is labeled the neuron, purple color labels the immune cell, and those red colors are those like mesh electronics. You can see that over this kind of months of the implantation, there's a minimal degradation or proliferation of the immune cells and the neurons in this like implantation site. With those kind of stable interface, we can capture those electrical activities, those single unit action potential stably from the behaving animal. So this is a continuous 13 months of the recording. You can see that the single unit action potential extracted from each electrode shows this remarkable stability that is over this 13 months of the recording. But to further test if it's indeed coming from the same neuron over the time, we need to design this like a high density electrical array that allow one neuron activity that can be simultaneously captured by the multiple electrical array, because the distance between the neurons to the different electrode is different. Therefore, they show this kind of distribution of the amplitude variations across a multiple electrical array. that give us the ability to test that it's not just the single unit action potential waveform is stable, but their distribution across multiple electrical array are also stably captured from the brain. That suggests it's coming from the same neurons. And be more specific, we can test some of those behavior-dependent neurons. For example, there's in the visual system, there's this neuron called the orientation-specific neuron that you can send this dynamic greeting to those like animals to allow the animal to watch those dynamic greeting, and this greeting can be like sent into the different angles. And there's like a 10%, less than 10% of the neuron will show this like a orientation-specific activity. We call it a neuron representation. That is a firing rate of the action potential difference when you have the different angles that the different dynamic greeting with the different angles sent back to the visual system. You can say that their neuron representation to the different angle is also stable. So this is the strong evidence that suggests that we can capture the activity stably from those neural systems, and most importantly, stably from the same neuron in the behaving animals. So this is the first part of our research that we try to build with the tissue-like nanoelectronics to significantly extend the temporal coverage of those kind of tissue interface. Then the second question is that how we can distribute a large number of the sensors across a 3D volume of the brain, right? Then this is currently like a solution from the industry, for example, the Neuralink, that they adopt this kind of previously developed the flexible tissue level flexible electronics that implant large number of the brain probe into the brain. Why is that? Because to enable the tissue level flexibility by using those rigid photolithographic compatible material, you need to make the device extremely thin. That will limit the number of the electrode actually you can incorporate into each brain probe. So for each brain probe, you only have like 8 to 16 electrode. Then when you want to implant like over thousands of the electrode across a 3D volume of the brain, you need to implant like over 100 brain probe. So that's why they need to have this kind of automatic robotics for the implantation. The ideal solution is not like that. It's like there's a lot of like regions on the brain probe that has been wasted. We want to have each brain probe like maybe contain thousands to more like 10,000 of the electrode, like a soft and flexible neural pixel like a probe, so that only few of the probe can cover a large number of the neurons across a 3D brain tissue for the long-term stable recording. However, the challenge is that, well, those like metals, those conducting material that can be made into a very thin structure, like a 20 nanometer, 40 nanometer, that is sufficient enough for their like a performance. But the dielectric layers that are used to encapsulate those conducting material, conducting structure cannot be further reduced. The previously, like we reduced the thickness of the isotope down to the like 800 nanometer in total for the both sides. That will be 400 nanometer on each side. That already approached to the limitations of the dielectric material. If you go further, smaller size or thickness, that will introduce this crosstalk or leakage current building in those like a system. So the real solution is that those dielectric material need to be made into like a soft structure, because that can allow you to incorporate more like a soft dielectric material without sacrifice this kind of mechanical flexibility of the entire probe. So the solution is that we want to bring those soft material into the bioelectronics fabrication. But for the soft material, there's several limitations for that. The first one is that why a material is soft or stretchable? Because its glass transition temperature is lower than the room temperature. So you can consider them as cross-linked liquid at room temperature, or they have like a large microforce structure within their like a network. Then they are very easily to be swelled by the organic solvent or sometimes even water. But for the multi-layer nanofabrication, for those high-density electronics, you always need to be like adopt this multi-layer fabrication process. A lot of different organic and water solvent need to use, so that you need to have those soft materials that have a strong solvent resistant, chemically orthogonal to all the other material for the other layers and also photopatternability. The second one is that we found that is mainly like done by my student Paul, that he tested so many different kind of elastomers, those soft material in the biofluidics. We found that the ion can also easily penetrate into those soft material. So that for those dielectric material over the time in those like a warm, salty water, they'll become ion conductor. Their impedance will drop significantly so that they will lose the ability to further encapsulate the electronics. So to solve that, we also need to have this long-lived elastomer in the physiological condition. So imagine that a material, a soft material, that need to strongly repel both like water, oil, ionic ions in the solution. That from the chemistry perspective is like this is introduced to me by Jonan, that it seems like only the perforated elastomer can work. So when I was a postdoc here, that Jonan introduced me to this perforated elastomer, but at that time, we want to use that as a dielectric layer for those stretchable transistors. And then later, my student found that it seems to be an ideal candidate for this soft dielectric layer for the brain probe, because we test its like a performance in the biofluidics over this 500 days testing. They show this remarkable stability, dielectric stability in the PBS solution compatible to the SU8. Actually, this is we in that paper we tested so many material, and this PFB is the only one that can support this kind of long-term like stability in the biofluidics. And because it's an elastomer, so we can tune its like a molecular weight to make it as soft as the brain. And then we further engineering it to the photoresist. Although this, because this is using the acrylate for the cross link, so it is very sensitive to the oxygen. When we need to really adapt it for the multi-layer nanofabrication, we need to fabricate them in the nitrogen environment. So this is a kind of a tricky step, but we make this kind of photoresist, and using that to do the multi-layer fabrication, have this like over 128 electrode fully integrated into one brain probe, and the brain probe still maintain the brain level mechanical property. So then the next story is very simple. We can implant them into the brain over a long time. They introduce a minimal immune response. We can use this high density electrical array, capture the action potential from the same neurons over the long time. But you see, this technology is a scalable technology. They have the potential to incorporate even more sensors, like not just like a few hundreds of sensors, but thousands to millions of sensors into the brain probe and still maintain the tissue level mechanical property. But by doing that, it's really hard to imagine, right? As a student, to fabricate 1 million sensors in the clean room at the school. And I'm sure no one here wants to do this type of a PhD study, right? So that we have to incorporate the company. We have to adopt those kind of semiconductor industry foundries for this kind of scalable fabrication. So, and also design the signal processing chip and the processing software. So this is currently we make it commercial available like a photoresist, and then we can do this large scale automatic multi-layer neural probe fabrication. We make it like a compatible with the rodents. You can see this, we can capture this freely behaving, we can capture the activity from the freely behaving animal over the long time, like constantly track the activity from the same neurons. We also, with the TMSA, build those like a signaling processing chip right now can support like over 4,000 of the channels. And in the future that we can further improve those technology and currently pursue this kind of a human clinical trial, test its real like applicability in the clinical application. But that is just one direction, right? We try to use the industry's capability, the brutal force to solve this problem. But then scientifically, there's another challenge when you want to distribute the sensor across the entire 3D volume of the brain, is that this is the EM imaging, reconstructed imaging of a neural structure. This structure is only 20 times 20 times 40 micrometers. And within this tiny region, you can see there's like a cellular structure from more than 100 different cells fully interwoven together. So when the brains get functionally mature, those neurons are connected with each other at a nanometer resolution. That is, no matter how soft or how flexible you design the probe, as long as you implant them into a matured brain tissue, you will break a certain neural connections. This kind of acute damage can now be avoided. So how to address this? So we found actually nature already provide us some solution. That is, over this course of the development, 3D brain tissue is really coming from a 2D stem cell sheet named the neural plate on the embryo. So this neural plate will first fold into a neural tube, and with further growth and folding to fold into a 3D brain tissue. So we are thinking that instead of just make the tissue level flexible electronics, if we can make a stretchable mesh-like electronics, integrate this stretchable mesh with the neural plate. This neural plate to neural tube folding and further expansion can help us distribute the sensors across the entire 3D volume of the brain. So we started to test this idea on the in vitro organoids, because the organoids is follow the same kind of in vivo developmental process. Like you have the stem cell sheet, they are gonna start to fold into like a ball-like structure, and after a certain development, they can grow into a 3D tissue. We are thinking to build this stretchable mesh electronics integrate with the 2D stem cell sheet, and when they grow into 3D organoids, they can self-distribute the electrons across the 3D tissue to test if this idea can be applied to the in vivo setting. So this is the initial idea. It's very simple. We just adopt those serpentine-like interconnects with our mesh electronics that are previously developed by Professor John Rogers' group, right? And you can see how stretchable they are. They can be released from the substrate into water. Even floating with water can easily stretch them. So we integrate them with the stem cell sheet. The stem cell sheet will first, due to cell-cell interaction, gonna shrink into a high-density cell plate, and actually further package the electronics into a high-density manner. So it's very good for this kind of design. And then start to grow into this ball-like structure after 3 days of culture. And after 20 days of culture, they can grow into a 3D organoids with the electronics completely distributed across this 3D organoids. So that we call it a cyborg organoids technology. And then we can induce them into whatever organoids we want. We start with the cardio organoids. You can say that the fully integrated electronics can adapt to this contraction of the cardio organoids. And this seamless integration allow us to long-term stably track the activity across the 3D cardiac organoids with this high-density recording. And more importantly, because of the stability of the recording, they can allow us to even visualize this gradually evolved action potential from the cardiac organoids. Start with this slow calcium-like activity, gradually emerge the hyperpolarization, and ultimately develop this depolarization spike. We can also induce those organoids into those brain organoids with the electronics fully distributed across the brain organoids and capture when the action potential first generated from this human brain organoids. But with those testing, ultimately we want to demonstrate that it can be applied to the in vivo real living system, right? So we implant them into the different kind of animal models, but right now focus on the aquatic animals because their entire embryo development is in the solution so that we can easily get access to it. So we started this with the frog, and then later with the axolotus is a kind of a spatial animals that with every part of the body can regenerate by itself. You can see that we can design those electronics with all the sensors incorporated in this region that is on the neural plate. This neural plate fold into a neural tube will self-internalize this stretchable electronics into those into this embryo. And after almost 2 weeks of the growth, they can grow into a freely moving tadpole with the electronics fully embedded in their brain. So this is showing this we can continuously capture the electrical activity from the same embryo over time, and no one have done this before. And this result is very interesting. At the early development stage, when the neural plate is forming, they have this global synchronized activity across this entire neural plate, but very slow, but synchronized activity. And then they start to gradually decouple into those brain region specific activity. And after like a fully like a development into the tadpole, they generate this kind of cell specific action potential from those brain regions. So that gives us the ability to capture how those brain gradually developed from like a bunch of the random neural network into a very specialized brain regions for the functions. So by integrating those tissue level soft electronics and cyborg engineering, we hope to further pursue in the direction that we can distribute those sensors across the entire 3D volume of the brain and other tissues to understand their functionalities. The last piece is that how we integrate the cell type specificity into this platform. So over my career, I kind of dedicate to try to develop this kind of technology. Start with some optogenetics tools like stimulate neuron and then record the activity. Or at here with Renan and Carl, we try to engineer the neurons to grow a conducting polymer and connect with the electrode. But then when I start at Harvard, that are really interested in those technologies that is this imaging-based in situ single cell R sequencing technology and incorporate them into the flexible bioelectronics. And so I'm sure that maybe some of you are not very familiar with this technology. I want to briefly explain it. So how this kind of so-called in situ single cell R sequencing work? That if you have a piece of the tissue you want to do the measurement, you can have this piece of tissue cross-linked into the hydrogel network. And this hydrogel network will cross-link all the biomolecules in situ in this intact piece of the tissue. Then you can do this kind of using the detergents to remove all the phospholipid bilayer, make the tissue transparent and accessible to other large biomolecules. Then you can design the DNA probe that is can hybridize with the mRNA. And this DNA probe will have the piece of so-called unique gene identifier, which can be sequenced out later to identify this DNA probe's identity. And then you introduce those probes, the probe will hybridize with mRNA in situ in this intact piece of the tissue. And then you do multi-round of the imaging-based sequencing, you can read out those DNA identity and their location so that you can infer their mRNA identity and the location. Then we can go through a couple of processes, the computer vision-based cell segmentation, mRNA copy number calling. So you can get the information that for those kind of cell mapping with how many different type of mRNA copy number in each cells. Then you can just go through this standard bioinformatics process, do the dimension reduction and the cell clustering to give each cell the specific cell type information. And in the end, you can generate a piece of the tissue with cell type information fully unraveled. But compared to all the other genetic engineering technology that I just mentioned, the advantage for this technology is that you can simultaneously not just look at 4 genes, 10 genes, 40 genes, you look at thousands of genes at the same time, give the most accurate cell type information for each cell type. This is especially important for the neural system because there are so many different types of the cell fully interwoven together. Then the problem is that how we can link those kind of spatially resolved gene expression information with our electrical recording and control. Because the soft electronic have the ability that once you incorporate them into the tissue, they will fully grow together with the tissue as a piece of a network so that you can harvest them together with the soft electronics fully embedded. And then you can go through this kind of whole tissue clearing, the gene expression mapping. But the question is that which channel that we are looking at is unknown. So that we can use the photolithography methods to incorporate a piece of like, for example, SU8 structure that is with the fluorescence dye embedded, tell us the channel information that we want to correlate. So that after that, we can directly look at the gene expression information from the cell with the channel and integrate their electrical information together with the gene expression information. Another problem for this technology is that the in situ single cell R sequencing is a terminate technology. When you want to look at the gene expression at this scale, you have to sacrifice the tissue. Then the question is that how we do this live tissue gene expression, cell type specific recording and control. Then that is where this kind of data analysis really comes from. Once we have the electrical activity and the gene expression from the different part, we can integrate them into the integrate them together at a single cell resolution. Then we can use all those kind of AI model to build the prediction model from using one modality to predict another. So this is how we design the technology. You can see that this is the flexible electronics, the sensors with this kind of fluorescence, we call it a fluorescence electronic barcode, a piece of the SU8 that we incorporate. It can tell us the information of these sensors. And then this is go through this like a 5 round of the in situ sequencing, and this kind of fluorescence barcode tell us where the sensor is. And this each dot is basically labeled one MRA that we are looking at. So ultimately, we have this whole tissue that has been in situ sequenced, and we got their electrical activity, we have their gene expression, and fully integrate together. So this is for the cardiac tissue. For the neuron tissue, it's slightly challenging. That is because we need to triangulate the neuron locations, because we don't know which neuron that we are looking at. They are firing differently. But as we said, that we need to design this, we can design this high density electrical array that one neuron activity can simultaneously capture by multiple electrode. So based on their like a variance of the amplitude across the different electrode, we can get this like a relative location of the neuron to this electrical array, so that then read out the gene expression from that neurons and incorporate the gene expression information with their electrophysiological information. So this ultimately gave us this multimodality measurement of the system, seamlessly integrated the cell type specific information with the electrical information. So what this technology can really help us to understand the system, right? That is, for example, I give you like 3 examples. The first example that we can do the multimodal cell state identification. For example, over the time course of the development, as I just show you, the cell at the different development stage will show different kind of electrical activity. They will have a different gene expression activity. And for this kind of study, people really want to correlate their like a different development state with their real development time. But you can see that if you just use gene expression to define the cell state, at the early development state, it is very easy to be separate with the resting development state because the gene expression differences are so different. But at a later development stage, you cannot separate them at all because the gene expression among the different type of cells are so small that you cannot further separate them. Using electrophysiology to define the cell state, they can be defined into 2 big clusters. But within cluster, it's not separable. But if we can integrate both gene expression and the electrophysiology, they can be separated into a well-defined cluster, correspond well to their real development state. And more important, as I say, that this kind of multimodality information give us the ability to look at the covariance of this 2 modality, that how gene expression change affects the electrophysiology or vice versa. We can design a very simple single layer neural network, do this sparse reduced rank regression to unsupervised tell us the correlation between different electrophysiological features with their gene expression difference. So that can show us what kind of gene expression that change what kind of electrophysiological features. So that automatically give us the model, like a link the gene expression with the electrophysiology. This is very remarkable because for all the traditional biophysics model, you can only correlate the ion channel expression difference with the electrophysiology. But for this model, because it's a completely statistical model, it's a data-driven statistical model, we can incorporate so many other genes, such as structural or metabolic related genes that previously cannot be incorporated in this kind of model to predict electrophysiology. And after we learn this correlation between these two modalities, immediately it can allow us to do another kind of a prediction model, use one modality to predict another. So this is a very simple couple autoencoder based cross modality prediction model, allow us to use electrophysiology predict gene expression or vice versa. And this is important because, as I say, that gene expression measurement at this scale is terminal measurement. You need to sacrifice the tissue and read out the gene expression. But our electrophysiology, on the other hand, especially those flexible electronics, can be used to continuously measure the electrical activity from the same piece of the tissue so that we can put them into the model to directly predict or infer a molecular change in this tissue. And more importantly, there's so many kind of like disease-related gene. For example, this LMNA mutation, that is one single gene change causes a lot of like a gene pathway change in those like patients with cardiac disease. And we can look at these patient-specific genes and show how different they are with the control, with the healthy cells. And we can directly put them into the model. It can predict its abnormal electrical activity, which show remarkable similar to this like activity that you record from these patients. And in the future, we are doing this so-called in silico perturbation or digital perturbation to perturb those genes and find what is the best strategy that we can bring it back to the normal, to bring this like abnormal electrical activity back to the normal patients. And in this era of the AI, it's data is not just the data is important. The model that have been trained by the data is also important. I just want to show you one example that we do this explainable multitask learning to understand the model that have been trained by this multimodal data. So this is a unique model that have been trained by those multimodal data to optimize the cross-modality prediction. And after this model has been trained, we can directly dissect this model using the explainable learning to look at how those neurons in the artificial neural network connect with each other. And with this dissection, just one step, it can tell us in each specific type of cell, the correlation between gene expression marker, electrophysiological feature, and the morphology features, and give us a lot of information for other to understand how those kind of different features correlate with each other. So you can see that we build this tissue-like nanoelectronics, enable this scalable and long-term stable electro recording at a single cell resolution. And then we build this cyborg engineering and the tissue level soft electronics to distribute the electronics across the 3D tissue. We build this in situ, we call it in situ electrosequencing to link the cell type specific information with the electrical recording and control, and use the AI to bridge all those kind of data together. So we kind of already merge electronics with biology across all the scale, from the molecular through the cell to the tissue. So what we are working on right now by leveraging this platform technology. The first one that we are really building this flexible brain-computer interface. We think it's going to revolutionize the entire brain-computer interface field. So let me explain. So the current brain-computer interface field, the real challenge is coming from the signal instability at the front end. Because the probe are rigid, because it causes kind of a drift inside the brain, immune response from the brain. Every day, basically you record activity from completely different neurons, even if you can record the activity stably. So the field is focused on developing this so-called manifold-based stabilizer to stabilize the signal. What does this mean? So if you consider each neuron activity as one vector, then ultimately, depending on how many neurons that you record, they will form this kind of n-neuron high-dimensional space. But because the neurons also connect with each other, so they cannot random fire. They're firing. So ultimately, the output is not this not in this high dimensional space. It's always a low dimensional geometry in this high dimensional space. And this geometry is called neural manifold. And how those like neural dynamics change over time will be a trajectory on this manifold that called the neural trajectory. For certain brain regions, such as motor cortex, their output seems to always be stable over a long time. So that while you can record activity from the different neurons on the different day, but because their output will always project into the same manifold, so you have this ability to realign those trajectory or manifold over the time based on this assumption. But for our case, there's no such problem because we can constantly record activity from the same neuron. So their output is always stable. The neural trajectory is always stable, that we do not need to do this kind of manifold-based alignment. On the contrary, we can use the stable recording as a training data to directly train a neural network to become like animal-specific brain decoder. Why this is useful? Because in the reality, there's no 100% yield. You always have one channel that is broken or one neuron is just died for no reason. And this animal-specific decoder can quickly adjust themselves and then give a stable output. But this is not that important. More importantly, is that if you want to go beyond motor cortex, how you can do it? Have you ever think about why the current brain-computer interface always just using the motor cortex, just read out the motor-related information? It's because other brain region exists this phenomenon called the neural representational drift. What does this mean? Brain is actually a dynamic system. Its neural activity constantly changed and never stopped. For so many other brain regions, basically, I feel like except the motor cortex, all the other brain regions, even you just give a constant input or output, the neural activity constantly change. For example, if you ask an animal, a mice, to watch the same movie again and again and again, every day, the neural activity that represents this movie, called neural representation, is different. And this is not just a visual cortex. It almost exists into every single brain regions. So how to extend the brain-computer interface to other brain regions? If we want to have so many different applications, we need to conquer that. And our technology seems to be ideal to conquer that because we can make sure that we can track the activity from the same neuron based on their waveform and the waveform distribution across the electrode. And then we can look at their like a neural representation, their firing dynamics to the different like a input or different output, and look at their like a relationship. So this is how we do. Like we have the animal watch the same movie again and again, and then capture those electrical activity from the brain. And using those waveform stability to confirm that we can capture the activity from the same neuron. Then we look at the neuron firing dynamics to the same movie. You can see that this is 3 representative neurons from the visual cortex of the animal that have been watching the same movie continuously for 5 months, the same movie. And you can see that their neural representation definitely changed at a single cell level. And actually, statistically, they never stopped. They constantly change. Over like 5 months, they never stopped. Previously, people think this may be related to the learning. But learning, you will like gradually like stable. This never stopped. And this kind of change is only related to how many times you watch a movie rather than how the interval of the time that we measure the signal. Then this question comes back, what is stable? After all, we still watch the same movie, right? By looking at the same neuron population dynamics, that means the neural manifold or neural trajectory, we found that it's very interesting, although at the single cell level, the dynamics constantly change. At the population level, also constantly change. But we found it's actually just like a keep like a rotate or stretch in this high dimensional neural space. That they preserve their topology. That means that if, for example, initially they are donorship topology, they keep this donorship. The donor just like a stretch, just a donor just a rotate. So this is mathematically called affine transformation. It's a linear affine transformation that we can easily compensate that, so that we can design a brain like a brain machine network that uses the AI to easily compensate this affine transformation, and then we can get a statistically significant higher like a visual information decoding. So this is not just in the visual system. We believe this is exists in almost every single brain regions, that we can apply this kind of compensation, this affine transformation compensation across multiple brain regions to really extend the brain-computer interface to other behavior, other process. And then that will be related to one of my own like interesting. You keep thinking, why is the brain designed in this way, right? Why we watch the same movie, but our representation in our brain constantly change and never stopped? Then one day, like I talked to my colleagues, like Professor Jie at Minnesota Computer Science Department, and he told me that this kind of a phenomenon in the machine learning called the catastrophic forgetting. What does that mean? So in the machine learning, if you have a small network, and you ask it to identify, you train it to identify a cat. Over the time, they will learn how to identify a cat. Then you train it to a different task, for example, identify a dog. After they learn how to identify a dog, they quickly forget about the cat. So you always need to build a much larger network. Why we need to have a large model to do multitask learning? Then we are thinking that, is that if we allow those like knowledge that be learned by the machine learning, and also like the brain, they never stop. They do not learn a stable like a local minimum in the latent space, but constantly change the inside this network. Will it conquer this important catastrophic forgetting issue in the machine learning? Then we design a completely different type of neural network, but still based on the deep learning, based on deep learning. That way, in the deep learning, when you learn a task, you will ultimately have all the information localized in the loss landscape as a local minimum. But instead, we have a local minimum. As this local minimum constantly change, we introduce a noise to the system. Like the feature of the biological brain is that it's not very stable, right? You get an emotion very easily. That's the synapse connection is never be like a stable numbers. Then we introduce like a very simple Gaussian noise, allow those kind of latent space, latent dynamics constantly change. Then we build this network, we call it a drift net. Very simple idea. And then we use it to train a GPT-2 because we don't have money to train GPT-3 or beyond. We just train the GPT-2 and to learn the different kind of like a news information. For example, you can see that for the traditional stable network, after it learns the AG news and then learns the Amazon news review, it's gonna quickly forget about the AG news. But after we introduce this drift, a very simple idea, after they learn this new task, they will still maintain the previously learned knowledge. And maybe over the time, like when we go through the force, like a Yahoo answer, this like performance shows slightly variation, but they still maintain previously learned knowledge. And on the contrary, for the stable traditional neural network, they completely forget all those like previously learned tasks. So you see that is really the beautiful part of the work, that by design those flexible electronics that we can enable very stable recording from the same neuron. Imagine like previously that every day you record activity from different neurons, and those neuron dynamics also constantly change. You can actually not decouple this is so-called the brain intrinsic dynamics from this recording dynamics. Right now, at least we can fix the one. We can keep record activity from the same neuron and observe what is the brain intrinsic dynamics during any kind of behavior or learning or memory process of the brain. Ultimately, we design this kind of paradigm that through this flexible brain-computer interface, we can extract the stable neural activity and directly use the stable neural activity to train AI. And through this kind of like an animal performing the different task process to reprogram the AI directly by using the brain dynamics, so that we can either build the AI to better control the brain or by dissecting this AI network to understand the neural intrinsic dynamics and further applied it to our own like a machine learning network. So this is the one application I will do. And also surprisingly that we are doing something that I'm not very familiar with before, but obviously the funding agency is super enthusiastic about this, like the stem cell engineering. That in the stem cell field, like there's a very significant problem that people try to address is that how to understand when the stem cell got functional mature. Like when, for example, if you induce the stem cell into the cardiomyocyte, when the cardiomyocyte can show this functional maturation as the cells in the body. So this like a flexible electronics technology seems to be very easy to address that. We use one example. Previously people found that when you incorporate some endothelial cells with stem cell derived cardiomyocyte, the stem cell derived cardiomyocyte become like more functional matured. But people don't know why, and the hypothesis is that those endothelial cells will form some kind of vascular structure to enable the nutrition and the oxygen to diffuse across the 3D tissue. And when we apply our technology to that, we found it's completely opposite. Even at the very beginning, when the endothelial cell has been integrated together with the cardiomyocytes, they show this like a significant different kind of electrical waveform. And over the time, this electrical waveform like constantly show this more functional matured features than those like a cardiomyocyte alone. And this like a statistical result that show that these different features has been all be like a significantly improved. And then we can use those machine learning algorithms to do the dimension reduction and build such as a so-called pseudo time analysis. So each like a electrophysiology feature will be learned into a latent dynamics. And ultimately in the latent space, they will connect it into a trajectory. So that we can build this kind of quantitative like a measurement of this compare those complex electrical waveform into a continuously changed trajectory. And you can see that like when the cardiomyocytes has been integrated with the endothelial cells, even at the very beginning, they already show the significant shift of this pseudo time towards like a more functional mature regions. And this is the statistical significance. And as we say that we can look at their gene expression after the electrical measurement, and we can look at how those kind of cell type information has been changed and all this kind of signaling pathway that has been changed, and use those like a standard bioinformatics to inform us which cell type like contribute most to the different type of the cell. And the ultimately finding that after the cardiomyocyte has been integrated with endothelial cell, after the functional maturation has been tailored, then cardiomyocyte genetically become more like an endothelial cell, and endothelial cell genetically become more like a cardiomyocyte. And through this kind of like a cross cell, like a ligand receptor exchange. And also, but this byproduct of this study is that we can build this continuous like a trajectory, pseudo time trajectory to inform us the cardiomyocyte's functional maturation stage by looking at their like electrical data. And we successfully build the correlation between the electrophysiology and the gene expression so that it can have a better information of how the transcriptional state change over the time. Then we are thinking that this data is ideally for those reinforcement learning, because reinforcement learning is require this kind of information, give a very clear state, and then send the reward and look at how this reward like change the state of the cell. Then we can build up this kind of closed loop system, use electrical to record, use electrical to stimulation, and build like an AI engine to learn how to control this kind of stimulation. In the era of the AI, it's very interesting that one thing that we don't need to really understand the underlying mechanism. We just build this AI and then we tell this AI, we just want to accelerate the functional maturation process along this trajectory by tailor those electrical stimulation. Can you do it? And the result is quite a surprise that in the first couple of those kind of training, this is a very simple Bayes optimization process. It's the first couple of the cycle of the training, 5 cycles that probably include like 25 samples. I really don't know why my students really have the patience to test like 25 samples. And at this time, the AI just randomly explore across this stimulation space. Doesn't show any like a kind of like a trend. But after this like 5 cycles of study, this like exploration, they seem to converge. They seem to converge. They learned something from the system and tell us to stimulate the cardiomyocyte in that way based on this electrical input. And we still don't know why, but the result is extremely remarkable. You can say this is AI-driven stimulation, and this is using the previous published fixed stimulation protocol, and this is non-stimulation protocol. They show this. Just look at the waveform. They show this significant improve of the waveform, and the whole tissue has been more synchronized. The speed of propagation across the cardiomyocytes has been statistically significantly improved. With their like molecular alignment has also been significantly improved. So this is like what we want to do in the future. That is really the AI-driven stimulation from this kind of closed loop system to improve the functional maturation of the cardiomyocyte of the stem cell. And another direction that is very interesting, that is the stem cell-derived pancreatic islet. That's a pancreatic system. So because of all of our publications, then some of the people tell me that Professor Douglas Melton and his postdoc tell me that actually, you know, among all those stem cell derived tissue, which one that have been really used in the clinical trial, that is the pancreatic islets. And when they tell me, I even don't know where the pancreas is, you know. And also they inform me that those pancreas cells actually derived from the same progenitor cells as the neurons during the developmental stage. So that they think that you should be able to measure those action potential like activity from the pancreatic tissue. So we have done so. We integrate those electronics with the stem cell derived pancreas. That you can see that we can measure those beautiful action potential like activity from the pancreas cells. And we can use the spike sorting, like do the neural data analysis to sort, to get the cell cluster. And based on alpha and beta cells show completely opposite like a reaction to the different glucose concentration. We can use glucose thresholding to inform us which one is alpha cell and which one is the beta cell. And we can use that to continuously monitor when the alpha and the beta cell has been shown this functional matured. And actually in the reality, we found like the alpha and the beta cells can be separated into more clusters. Like some of them show a strong response to the glucose, some show like a weak response to the glucose stimulation. And ultimately, their functional maturation is not the total number of alpha beta cells have been changed, but this ratio between the different type of alpha beta cells has been changed over time. And we can train those like stem cell derived pancreatic islet, let them to show the different kind of behavior to different glucose like a stimulus, and ultimately train them really function like a pancreatic tissue in our body, have this rhythm like show that response to different meal cycles in the in vitro setting. And of course, like we can fix them and use the gene expression to tell us which kind of gene change actually govern this. And ultimately, we want to build this, combine all the technology together to build this functional brain cell atlas. Like with my colleagues that we already using the in situ single cell RNA sequencing to map every single cell's gene expression across the 3D brain tissues. And use those gene expression, we can redefine the brain anatomy. Can you imagine actually brain anatomy? The Allen Brain Atlas is actually defined by using the methods that have been developed 100 years ago. And there's for so many brain regions, there's no ground truth. And by using the gene expression, single cell gene expression, we can redefine the brain anatomy. Actually, we find so many different brain regions that was not reported before. And certainly, this is just like like a brain initiative work. So that last year we have a 10 paper published on the Nature at the same time. And then one of my students, very good at those data analysis and build this foundation model by leveraging the single cell gene expression and their spatial information together to integrate everybody's data into a huge like a brain cell atlas contain more than 18 millions of the cell. And this brain cell atlas actually can also incorporate other modality into this cell atlas through this kind of a spatially resolved information, gene expression information. So this is what we are doing. We are further extended this in situ electrosequencing. We implant those probe to those brain regions that haven't been identified before, then we look at what is the unique behavior-dependent electrical activity. And after that, we sacrifice the tissue, look at the gene expression, and use those kind of correlated data. We can map it back to this big centralized atlas. And as I said, like nowadays, it's not just the data that is important, but the network that has been trained by this data are very important. That we are training this large model and then using this explainable learning to dissect the model to tell us the correlation between the electrical activity and the cell type information with the animal behavior. So we hope that in the next 5 years, we can build this Google map of the brain, this brain functional atlas that guide us where we can record or like stimulate the brain. So that's all my talk. And in the end, I want to disclose my conflict of interest. And all the research has been done by my student and great collaborators. And we received the funding agency, fundings from those funding agency. Thank you. Happy to answer any question.
Wow. Thank you so much. Amazing research, Dr. Liu. Yes, and I wanted to get to a couple of questions online so we can close out the webinar part.
Sure.
There were, and maybe we can send some more to you.
Sure.
Okay. About the density of the probes, there was big interest in being able to have up to 1 million channels.
Yeah.
And so there's one question about, well, what do you hope to get by having so many channels?
Yeah. I think the first one is that right now the Brain Initiative has finished. So to understand brain, actually we need to have a 3 mapping: cell connectivity mapping, cell gene expression mapping, and the cell electrical activity mapping. Currently, the cell gene expression mapping has almost been finished by the Brain Initiative. We are also part of that. Right now, they are working on the cell connectivity mapping, and we already have the methods, and we are supposed to finish that in next 5 to 10 years. But for the electrical mapping, we still have no methods at all. And by not just millions, if we can just go 10,000s, a reliably recording of the neural activity over the different kind of scenario, we will have the chance to build this kind of activity mapping. And ultimately, those data need to be incorporated together. So from my perspective, although there are so many other things that we can open up, but this will be the most critical thing that we need to do as soon as possible.
Great. And you're solving many challenges in order to be able to get there, such as addressing drift. Are there other bottlenecks that you think are not clear how to solve them to get to the mapping you want to do?
Yeah, I think in terms of the drift, right now we found this in the visual cortex that is seems to have this linear fine transformation can explain those drift, but there may be some nonlinear process that's involved, especially that if we reach some regions there's those dopamine, those more like a chemical information rather than just the electrical information govern the process. Then there's so many a nonlinear process that need to be understand. And in addition, that by measure this chemical information itself is very critical for us to better understand how those neural representation change. And for our technology, we are very limited just by the electrical information, so that we also need to further incorporate at least the chemical sensors into the platform. Yeah.
Wonderful. Well, thank you. Let's take a couple questions from the room, and then we can have more informal discussion. Sure. Professor Bao.
So many different facets together. The core of your work is the stable recording of the same neuron. And so is just being soft the only reason you can do that, or there are other factors that enable you to have the stable recording?
I think so. For example, if you use a neural pixel, we also analyze the neural pixel data. Your neural pixel is not like you cannot have a stable recording. It's just they drift so that they only have a small portion of the neurons that is stable. If you a computational power is strong enough, you always can extract those stable neurons. By change them into a flexible, we found that the percentage of the stable neuron that we can record has been substantially increased.
Is there a quantitative comparison that you have published to compare like your data, how much it drift or not drift at all compared to some rigid probe?
It's very interesting. This field, like people do not share data. So we actually try to find some like published data from other people, but because people don't share raw data, we need a raw data to quantify that. We need to look at the waveform. Most of the data people share are just a spiking data. They only have for each spike, they have a spike number here. So there's no like a waveform information at all. So that we don't have the data that can allow us to make this comparison at this moment.
Then how about the NeuroPixels?
Yeah, NeuroPixel, they share data that is the spike data. They don't share the raw data.
I see.
Yeah. Okay. So we are very interested to look at the data, for example, released by industry, like a Neuralink, or by the, although precision neuroscience don't do that, but we're looking forward to other people's like recording data and do the systematic comparison. And if anyone that's interested, we also are willing to like very, very much want to do that, because there's some editors already talked to me that they would like to look at this kind of cross-technology comparison. Yeah, but we are limited by the data.
Okay. Yeah, I have other questions, but I let the other people ask.
Right. No, nice, very nice talk. You mentioned you just mentioned the chemical route. Do you think that should be studied like concurrently with the electrical signaling, or like separate? I mean, it's all complementary, but what do you think are some of the not best, but perhaps manageable ways of studying that and incorporating that if it's ready to be done? So.
Yeah, that's a very good question. I think it needs to be started together. So previously, I'm not familiar with that, but the more time that I'm getting into this field, I found for this type of study, the integrative methods is always most useful. That means that you have a different modality incorporate together to measure from the same animal, same brain under the same like behavior. And this correlative integrative like a measurement is always the data are always most valuable. Yeah. So if we can, we want to study them together, rather than separately. Yeah.
That's making a hard job even harder though.
Yeah, I mean, even harder. So there's alternatives that we try to train like a so-called so it's kind of a cliche now because everybody is doing that, but it's actually very useful that train the foundation model and then use the foundation model to bridge the different kind of modality. Yeah.
Okay, this is great. I see more hands in the room. Let's close out the formal part of our lecture now at the UR seminar, and wish everybody happy holidays. And we look forward to seeing you at our next event in the new year. And let's thank Dr. Jialu one more time.